Hello everyone, I am Pushpangna from IIT Kanpur and I am a Prime Minister Research Fellow in the May 2020 lateral entry cycle. This lecture video is going to focus on Coronaviridae. The coronaviruses are the largest group within the nidovirales. In order, it is an order that comprises the families Coronaviridae, Arteriviridae, and Roniviridae. The arteriviruses, a small group of mammalian pathogens, the roniviruses, which infect shrimp, and a very recently isolated mosquito-borne virus, which is not yet classified, are currently the only members of the order having invertebrate hosts. Nidoviruses are membrane-enveloped, non-segmented, positive strand RNA viruses that are set apart from other RNA viruses by certain distinctive characteristics. Their most significant common features are: first, an invariant general genomic organization with a large replicase gene upstream of the structural protein genes. Second, the expression of the replicase transcriptase polyprotein by means of ribosomal frame shifting. And third, a collection of unique enzymatic activities contained within the replicase transcriptase protein products. And fourth, the expression of downstream genes via transcription of multiple 3' nested subgenomic messenger RNAs or mRNAs. This last property has provided the name for the order, which comes from Latin nido, for nest. It should be noted that the replicative similarities among the three nidovirus families are offset by marked differences in the numbers, types, and sizes of their structural proteins and great variation among the morphologies of their virions and nucleocapsids. Coronaviruses are now classified as one of the two subfamilies Coronavirinae in the family Coronaviridae. The other subfamily, Torovirinae, includes the toroviruses, which are pathogens of cattle, horses, and swine. This lecture will concentrate exclusively on the Coronavirinae. Coronaviruses have long been sorted into three groups. Originally, on the basis of serologic relationships and subsequently on the basis of phylogenetic clustering. Following proposals that were recently ratified by the International Committee on Taxonomy of Viruses, or the ICTV, these groups, the alpha, beta, and gamma coronaviruses, have now been accorded a taxonomic status of genera. The ICTV classification have also established rigorous criteria for coronavirus species definitions in a manner consistent with those used for other viral families. As a consequence, some viruses previously considered to be separate species are currently recognized as a single species. For example, the viruses now grouped within alpha coronavirus or beta coronavirus. Additionally, the new classification criteria resolves any previous uncertainty about the taxonomic assignment of the virus that causes SARS or severe acute respiratory syndrome as a beta coronavirus. Almost all alpha and beta coronaviruses have mammalian hosts. In contrast, the gamma coronaviruses, with a single exception, have been isolated from avian hosts. The focus on these viruses came about largely because they were amenable to isolation and growth in tissue culture. However, since 2004, molecular surveillance and genomic efforts initiated in the wake of the SARS epidemic have led to discovery of a multitude of previously unknown coronaviruses that now constitute most members of this subfamily. Notably, most of the newly recognized species are identified in bats, which constitute one of the largest orders within the mammals. Diverse coronaviruses have been described from bats, principally in Asia, but also in Africa, Europe, and North and South America. These viruses include likely predecessors of SARS-CoV, but also 4 unique species of alpha coronaviruses and 3 species of beta coronaviruses. Birds have also proven to be a rich source of new viruses. Novel avian coronaviruses have been found to infect geese, pigeons, and ducks, and highly divergent coronaviruses recently identified in bulbuls, thrushes, and manas have the potential to gain a fourth genus in the Coronaviridae. It has been proposed that bats and birds are ideally suited as reservoirs for the incubation and evolution of coronaviruses owing to their common ability and their propensity to roost and lock. The most categorically harmful of human coronaviruses is SARS-CoV, now SARS-CoV-2. There are other human coronaviruses as well, which are designated HCOVs. The first is alpha coronavirus, HCOV 229E and HCOV NL63, and the beta coronaviruses HCOV OC43 and HCOV HKU1 typically cause common cold. Remarkably, HCOV NL63 and HCOV HKU1 were only discovered recently in the past. Although generally associated with upper respiratory tract infections, the extant HCOVs can also cause lower respiratory tract infections and have more serious consequences in the young, the elderly, and the immunocompromised individuals. In particular, HCOV NL63 is strongly associated with childhood groups, and the most severe HCOV HKU1, OC43, and 229E infections are manifest in patients with other underlying illnesses. Virions of coronaviruses are roughly spherical and exhibit a moderate degree of pleomorphism. In the earlier literature, viral particles were reported to have average diameters of 80 to 120 nanometer, but were far from uniform with extreme sizes from 50 to 200 nanometer. The spikes of coronaviruses, typically described as club-like or petal-shaped, emerge from the virion surface as stalks with bulb-like distal termini. Some of the variation in particle size and shape was likely attributable to stresses exerted by virion purification or distortions introduced by negative staining of samples for electron microscopy. More recent studies employing cryo-electron microscopy and cryo-electron tomography have produced images in which virion size and shape are far more regular, although still pleomorphic. These studies, which examined a number of alpha and beta coronaviruses, converge on mean particle diameters of 118 to 136 nanometer, including the contributions of the spike, which project some 16 to 20 nanometer from the virion envelope. Enclosed within the virion envelope is the nucleocapsid, a ribonucleoprotein that contains the viral genome. The structure of this component is relatively obscure in images of whole virions. However, its makeup has been partially displayed by electron micrographs of spontaneously disrupted virions or of virions solubilized with non-ionic detergents. Such studies revealed another distinguishing characteristic of coronaviruses. They have helically symmetrical nucleocapsids. Helical symmetry is common for negative strand RNA virus nucleocapsids, although it is highly unusual for positive strand RNA animal viruses, almost all of which have icosahedral capsids. The best resolved images of the coronavirus nucleocapsid, which were obtained with HCOV 229E, showed filamentous structures 9 to 13 nanometer in diameter with 3 to 4 nanometer wide central canals. These filaments were thinner and less sharply segmented than paramyxovirus nucleocapsids. However, widely ranging and sometimes discrepant parameters have been reported for the nucleocapsids of other coronaviruses, varying with both the viral species and the method of preparation. Thus, further work is needed to clearly delineate the diameter, symmetry, length, and protein RNA stoichiometry of this virion component in isolation. More recently, coronavirus ultrastructure studies have suggested that when packaged within the virion envelope, the helical nucleocapsid is quite flexible, forming coils and other structures that fold back on themselves. Coronaviruses contain a canonical set of four major structural proteins: the spike or S protein, membrane or M protein, and the envelope or E protein, all of which are located in the membrane envelope, and the nucleocapsid N protein, which is found in the ribonucleoprotein core. The distinct surface spike of coronaviruses are composed of trimers of S molecules. S is a class 1 viral fusion protein that binds to host cell receptors and mediates the earliest steps of infection. In some cases, S protein can also induce cell-cell fusion late in infection. The S monomer is a transmembrane protein of 128 to 160 kilodalton, composed of a very large N-terminal ectodomain and a tiny C-terminal endodomain. This protein is inserted via a cleaved signal peptide into the endoplasmic reticulum or ER, where it obtains end-linked glycosylation, increasing its mass by some 40 kilodalton. Comprehensive mapping of glycosylation sites has not been carried out for any S protein. However, an analysis of the SARS-CoV S protein showed that at least half of its candidate sites are glycosylated. The early steps of glycosylation occur co-translationally, and this modification assists monomer folding and proper oligomerization. Terminal glycosylation is then completed subsequent to trimerization. S-protein monomer folding is also accompanied by the subset of the numerous cysteine residues of the ectodomain. The positions of S-protein cysteines are well conserved in each coronavirus genus. Disulfide linkages have yet to be mapped. In many beta and gamma coronaviruses, for example, mouse hepatitis virus MHV, bovine coronavirus BCOV, and infectious bronchitis virus IBV, the S protein is partially or completely cleaved by a furin-like host cell protease into two polypeptides, denoted S1 and S2, which are roughly equal in size. Correspondingly, in coronaviruses that do not have detectably cleaved mature S proteins, the N-terminal and C-terminal halves of the molecule are also designated S1 and S2 respectively. S-protein cleavage occurs immediately downstream of a highly basic pentapeptide motif, and the extent of proteolysis correlates with the number of positively charged residues in the motif. The S1 domain is extremely variable, exhibiting very low homology across the three genera and often diverging extensively among different isolates of a single coronavirus. By contrast, the S2 domain is highly conserved. For those coronavirus in which it occurs, S1-S2 cleavage is a late event in virion assembly and release from infected cells. For many other coronaviruses, an alternative type of S protein cleavage or the S2- cleavage takes place during the initiation of infection, activating the molecule for fusion. The differing functions of S1 and S2 and the role of proteolysis will be discussed in the later slides. A complete high-resolution structure has been determined for the coronavirus S protein. All currently available structural and biochemical evidence accords well with an early proposal that S is functionally analogous to the influenza HA protein. In this model, the S1 domain of the S protein oligomer make up the bulbous receptor binding portion of the spike. The narrow stalk of the spike distancing the bulb from the membrane is a coiled coil structure formed by association of the heptad repeat regions HR1 and HR2 of the S2 domains of monomers. The most abundant structural protein in coronavirus, the membrane or the M protein, gives the virion envelope its shape. The M monomer, which ranges from 25 to 30 kilodalton, is a polytopic membrane protein that is embedded in the envelope by three transmembrane domains. At its amino terminus is a very small ectodomain. The C-terminal ectodomain of M accounts for the major part of the molecule and is situated in the interior of the virion or on the cytoplasmic face of intracellular membrane. Although it is inserted co-translationally into the ER membrane, The M protein generally does not bear an amino terminal signal peptide. For IBV and MHV, either the first or the third transmembrane domain of M alone suffices as a signal for insertion and anchoring of the protein in its native membrane orientation. Anonymously, M proteins of the alpha coronavirus 1 species do contain cleavable N-terminal signal peptides, although it is not clear whether these are necessary for membrane insertion. The ectodomain of M is modified by glycosylation, which is usually N-linked. However, a subset of beta coronavirus M proteins exhibit O-linked glycosylation, and the MHV M protein has served as a model for this study of post-translational modification. Glycosylation of M influences both organ tropism and the interferon or IFN inducing capacity of some coronaviruses. M proteins are moderately well conserved within each coronavirus genus but diverge considerably across genera. The most variable part of the molecule is the ectodomain. By contrast, a short segment overlapping in the third transmembrane domain and the start of the endodomain exhibits a high degree of sequence conservation that is seen even in torovirus M proteins. Like most multi-spanning membrane proteins, the M protein has been refractory to crystallization. However, recent cryo-electron microscopic and tomographic reconstructions have provided a glimpse of the structure of this protein within the virion envelope. These studies reveal that the large carboxy terminus of the M extends some 6 to 8 nanometer in the viral particle and is compressed into a globular domain, consistent with early work showing that the endodomain is very resistant to proteases. The observed M structures are likely to be dimers, the monomers of which are associated through multiple interacting regions. M dimers appear to adopt two different conformations, a compact form that promotes greater membrane curvature and a more elongated form that contacts the nucleocapsid. The E protein is a small polypeptide of 8 to 12 kilodalton that is found in limited amounts in the virion envelope. Despite its major presence, no wild-type coronavirus has been discovered to lack this protein. Engineered knockout or deletion of the E gene has effects ranging from moderate to severe to lethal. Thus, although E is not always essential, it is critical for coronavirus infectivity. E protein sequences are widely divergent even among closely related coronaviruses. However, all E proteins share a common architecture, a short hydrophobic amino terminus followed by a large hydrophobic region, and lastly, a large hydrophilic C-terminal tail. E is an integral membrane protein, but it does not have a cleavable signal peptide and is not glycosylated. Beta and gamma coronavirus E proteins are palmitoylated on cysteine residues downstream and adjacent to the hydrophobic region. This modification remains to be found in an alpha coronavirus E protein. The membrane topology of E is not completely resolved. Most evidence indicates that this polypeptide transits the membrane once with an N-terminal ixodomain and a C-terminal endodomain. Residing in the interior of the virion, the N protein is the sole protein constituent of the helical nucleocapsid. Monomers of this 43 to 50 kilodalton protein bind along the RNA genome in a beads on a string configuration, common to other helical viral nucleocapsids. However, unlike the nucleoproteins of rhabdo and paramyxoviruses, the coronavirus N protein provides little or no protection for its genome against the action of the ribonucleases. The bulk of the N protein monomer is made up of two independently folding domains designated the N-terminal domain or NTD and C-terminal domain or CTD, although neither includes its respective terminus of the N molecule. Crystal or solution structures have been determined for the NTDs and CTDs of SARS-CoV, IBV and MHV. Flanking the NTD and CTD are three spacer elements, the central one of which contains a serine and arginine-rich tract, or the SR region, which was noted to reassemble the SR domains of RNA splicing factors. Another functionally distinct region of N, the carboxy terminal domain, has been designed genetically. The spacer segments and domain 3 are each likely to be intrinsically disordered polypeptides. Most of the N molecule, including the NTD and CTD, is highly basic. By contrast, domain 3 is acidic. There is only one moderate degree of sequence homology among N proteins across the three genera, with the exception of a stretch of 30 amino acids within the NTD that is highly conserved among all coronaviruses. N protein is a phosphoprotein modified at a limited number of serine and threonine residues. Phosphorylation sites have been mapped for a representative coronavirus for each genus, and targeted sites collectively fall in every domain and spacer region of the N molecule. Thus, a general pattern for N protein phosphorylation cannot yet be discerned, nor have all responsible kinases been identified, although there is evidence linking glycogen synthase kinase 3 to phosphorylation of the SR region. The role of phosphorylation is also not known but is thought to have regulatory impact. Phosphorylation has been suggested to trigger a conformational change in N protein, and it may enhance the affinity of N for viral versus non-viral RNA. The most conspicuous function of the N protein is to bind to viral RNA. Nucleocapsid formation must involve both sequence-specific and non-specific modes of RNA binding. Specific RNA substrates that have been identified for N protein include the transcription regulating sequence and the genomic RNA packaging signal. The NTD and CTD are each separately capable of binding to RNA ligands in vitro, and the structures of these domains offer some clue as to how this is accomplished. The NTD consists of a U-shaped B platform with an extruding beta hairpin, which presents a putative RNA binding groove rich in basic and aromatic amino acid residues. The CTD forms a tightly interconnected dimer which exhibits a potential RNA binding groove lined by basic alpha helices. Some work suggests that in the intact N protein, optimal RNA binding requires concerted contributions from both the NTD and the CTD. A significant fraction of nucleocapsid stability also results from interactions among N monomers. This level of association is generally attributed to the CTD. However, additional regions of NN interactions have been mapped to the NTD and to domain 3. Another crucial function of N protein is to bind to M protein. This capability is provided by domain 3 of N protein. A fifth prominent structural protein, the hemagglutinin esterase or HE protein, is found in only a subset of the beta coronaviruses, including murine coronaviruses, beta coronavirus 1 and HCOV HKU1. In virions of these species, HE forms a secondary set of short projections of 5 to 10 nanometer arrayed beneath the canopy of S protein spikes. The 48 kilodalton He monomer is composed almost entirely of an N-terminal ectodomain. This is followed by a transmembrane anchor and a very short C-terminal endodomain. He is inserted into the ER by means of a cleave signal peptide and acquires an additional 17 kilodalton of N-linked glycosylation at multiple sites. The assembled protein is a homodimer, the subunits of which are connected by disulfide bonds. As its name indicates, the He protein contains a pair of associated activities. First, it is a hemagglutinin, that is, it has the capability to bind to sialic acid moieties found on cell surface glycoproteins and glycolipids. Second, He exhibits acetylesterase activity with the specificity for either 9O or 4O acetylated sialic acids. These characteristics are thought to allow He to act as a cofactor for S protein, assisting attachment of virus to host cells as well as expediting the travel of virus through the extracellular mucosa. Consistent with this notion, the presence of He in MHV dramatically enhances neurovirulence in the mouse host. Conversely, the He protein is a burden to the virus in tissue culture, where its expression is rapidly counter-selected. The two activities of the He protein are strikingly similar to the receptor binding and receptor destroying activities found in influenza C virus. And remarkably, the coronavirus He gene is clearly related to the influenza C HEF gene. However, toroviruses also possess a homologue of the He gene, raising the possibility that all three of these virus groups evolved from a common ancestor. This kinship is further corroborated by the crystal structure of the beta coronavirus He protein, which reveals separate receptor binding and acetylesterase domains perched atop a truncated membrane proximal region. The He protein thus resembles a squat version of its influenza virus counterpart, shortened because it lacks the fusion domain stalk of the HEF protein. The coronavirus genome, which ranges from 26 to 32 kilobase pair, is the largest among all RNA viruses, including RNA viruses that have segmented genomes. This exceptional RNA molecule acts in at least three capacities: as the initial mRNA of the infectious cycle, as the template for RNA replication and transcription, and as the substrate for packaging into progeny viruses. Assembly consistent with its role as an mRNA, the coronavirus genome has a standard eukaryotic 5' terminal cap structure and a 3' polyadenylate tail. The genome comprises a basic set of genes in the invariant order 5' replicase SEMN 3'. With the huge replicase gene occupying 2/3 of the available coding capacity. The replicase transcriptase is the only protein translated from the genome. The products of all downstream open reading frames, ORFs, are derived from subgenomic mRNAs. The 5' most position of the replicase gene is dictated by the requirement for expression of the replicase to set in motion all subsequent events of infection. The organization of the other basic genes, however, does not seem to reflect any underlying principle, because engineered arrangement of the downstream gene order is completely tolerated. Dispersed among the basic genes in the 3' most third of the genome, There are from 1 to as many as 8 additional ORFs which are designated accessory genes. These can fall in any of the intergenic intervals downstream of the replicase gene, except, curiously, never between the E and the M genes. In some cases, an accessory gene can be partially or entirely embedded as an alternate reading frame within another gene, for example, the internal gene of MHV or the 3B gene of SARS-CoV. Accessory genes are generally numbered according to the smallest transcript in which they fall. Consequently, there is usually no relatedness among identically named accessory genes in coronaviruses of different genera, such as the 3A genes of SARS-CoV, feline coronavirus, and IBV. Some of these extra ORFs are thought to have been acquired through ancestral recombination with RNA from cellular or heterologous viral sources. The HE gene is the best supported example of this type of horizontal genetic transfer. Two other such candidates are the 2A gene found in murine coronavirus and beta coronavirus 1, which encodes a putative 2' to 3' cyclic phosphodiesterase, and gene 10 of beluga whale coronavirus, which encodes a putative uridine cytidine kinase. Notably, the 2A gene has a homologue embedded as a module within the replicase gene of the torovirus, which is a situation also consistent with the horizontal transfer. The origin of most accessory genes, however, remains an open question. It is plausible that some of them evolved through intragenic recombination, resulting in gene duplication and subsequent divergence, as suggested for several of the accessory genes of SARS-CoV. Almost all accessory genes that have been examined are expressed during infection, although their functions are incompletely understood. The protein products of most accessory genes are non-structural. However, this rule is not without exception. The HE protein, the MHV1 protein, and the products of SARS-CoV ORFs 3A, 6, 7A, 7B, and 9B are all components of virions. Mutational knockout or deletion of accessory genes has revealed that none are essential for viral replication in tissue culture. Conversely, accessory gene ablation or transfer to another virus can have profound effects on viral pathogenesis. In some cases, the basis for this is understood to result from interactions with host innate immunity. Coronavirus infections are initiated by the binding of virions to cellular receptors. There then follows a series of events culminating in the delivery of the nucleocapsid to the cytoplasm, where the viral genome becomes available for translation. Individual coronaviruses usually infect only one or a few closely related hosts. The interaction between the viral S protein and its cognate receptor constitutes the principal determinant governing coronavirus host species range and tissue tropism. This has been convincingly shown in two ways. First, the expression of a particular receptor in non-permissive cells of a heterologous species renders those cells permissive for the corresponding coronavirus. Second, the engineered replacement of the S protein ectodomain changes the host cell species, specifically or tissue tropism of a coronavirus in the predictable fashion. The amino terminal, more variable half of the spike protein S1 is the part that binds to the receptor. Binding leads to conformational changes that result in fusion between virion and cell membranes, mediated by the more conserved half of the spike protein S2. The region of S1 that contacts the receptor the receptor binding domain or RBD varies among different coronaviruses. For MHV, the RBD maps to the N-terminal section of S1. By contrast, RBDs for SARS-CoV, HCoV NL63, transmissible gastroenteritis virus TGEV, and HCoV 229E34 fall in the middle or C-terminal sections of S1. The known cellular receptors for alpha and beta coronaviruses that have been known to date, and no receptors have been identified for gamma coronaviruses. The MHV receptor MCCAM1 was the first discovered coronavirus receptor, as well as one of the first receptors identified for any virus. That this molecule is the only biologically relevant receptor for MHV was made clear by the demonstration that homozygous CCAM1 knockout mice are totally resistant to infection by high doses of MHV. CCAM1 is a member of the carcinoembryonic antigen CEA family within the immunoglobulin Ig superfamily, and in its full-length form contains 4 Ig-like domains. A diversity of 2 and 4 Ig domain isoforms is generated by multiple alleles and alternative splicing variants of CCAM1. The wide range of phylogeneticity of MHV in mice is thought to be strongly affected by the interactions of S proteins of different virus strains with the array of receptor isoforms that are expressed in mice of different genetic backgrounds. Although their S proteins are phylogenetically very close to that of MHV, the beta coronaviruses BCoV and HCoV OC43 do not use CCAMs to infect their hosts. Rather, the only currently known attachment factor for these coronaviruses is an N-acetyl 9-O-acetylnuraminic acid. The recently solved structure of the MHV RBD complex with MCCAM1 has allowed the identification of key residues at the S protein receptor interface. Coupled with mutational analysis, this structure reveals why the S protein of BCoV and HCoV OC43 cannot bind the MHV receptor, and conversely, why MHV does not bind to bovine or human CCAMs. Most alpha coronaviruses use aminopeptidase N or APN of their respective host species as a receptor. APN, also called CD13, is a cell surface zinc binding protease that is resident in respiratory and enteric epithelia and in neural tissue. The APN molecule is a heavily glycosylated homodimer. Mutational and inhibitor studies have shown that its enzymatic activity is not required for viral attachment and entry. In general, the receptor activities of APN homologs are not interchangeable among species. However, feline aminopeptidase N or FAPN can serve as a receptor not only for the FIPV but also for canine coronavirus, TGEV, and HCoV 229E.567. This circumstance has been exploited for the construction of chimeric APN molecules to map the basis for receptor recognition. Such studies have found 3 small linearly discontinuous determinants in APN that govern the species specificity of this subgroup of alpha coronaviruses. The receptor for SARS-CoV, angiotensin converting enzyme 2 or AC2, was discovered with notable rapidity following the isolation of the virus. AC2 is a cell surface zinc binding carboxypeptidase involved in regulation of cardiac function and blood pressure. It is expressed in epithelial cells of the lung and the small intestine, which are the primary targets of SARS-CoV, as well as in heart, kidney, and other tissues. As with APN, the receptor role of AC2 appears to be independent of its enzymatic activity. Although the SARS-CoV S protein binds to the catalytic domain of AC2, active site mutation or chemical inhibition does not detectably affect the activity of AC2 to associate with S protein or to promote syncytia formation. The crystal structure of the SARS-CoV-2 S protein RBD in complex with AC2 shows the RBD cradling one lobe of the claw-like catalytic domain of its receptor. Remarkably, AC2 also serves as the receptor for the alpha coronavirus HCOV NL63, and the corresponding structure complex for that virus reveals that the HCOV NL63 RBD and the SARS-CoV RBD bind to the same motifs. Because the SARS-CoV and HCOV NL63 RBDs have neither sequence nor structural homology, this finding strongly supports the notion that they have independently evolved to bind to the same hotspot on the AC2 surface. Analysis of the SARS-CoV RBD-AC2 interface have additionally demonstrated the structural basis for the final jump of SARS-CoV from palm civets to human hosts. These studies found that merely 4 critical residues constitute the major species barrier between the civet and human AC2 molecules, and that mutation of only 2 key RBD residues was sufficient for civet SARS-CoV S protein to gain the ability to productively bind human AC2. The entry of the virions into cells results from large-scale rearrangements of the S protein that lead to fusion of viral and cellular membranes. These rearrangements are triggered by some combination of receptor binding, proteolytic cleavage of S, and exposure to acidic pH. The S proteins of many coronaviruses are uncleaved in mature virions and require an encounter with a protease at the entry step of infection to separate the receptor binding and fusion components of the spike. The details of proteolytic activation are still incompletely understood but have been best studied for SARS-CoV-2. In the cell types in which this virus is most commonly grown in tissue culture, viral entry depends on cathepsins, which are acid-activated endosomal proteases. The infectivity of SARS-CoV is thus suppressed by cathepsin inhibitors or by lysomotrophic agents. However, cell-bound SARS-CoV can alternatively be activated by treatment with extracellular proteases such as trypsin or elastase. This route of activation greatly enhances the infectivity of SARS-CoV and allows the virus to enter from the cell surface, thereby rendering the infection insensitive to lysomotrophic agents. The same pattern of proteolytic activation, cathepsin dependence, and its circumvention by exogenously added protease is observed with a particular strain of MHV, that is MHV2, that is unique in having an uncleaved S protein. The site of cleavage of receptor-bound SARS-CoV S protein by cathepsin or by exogenous trypsin differs from that of the S1-S2 cleavage, which occurs in other coronaviruses upon exit from the cell. Cleavage at entry takes place at a locus, that is at S2' within the S2 half of the molecule immediately upstream of the putative fusion peptide. It It is not yet clear if cleavage at analogous S2- sites is the pattern for all coronavirus S proteins. However, the emerging pattern is that proteolytic activation of S protein is required for infectivity, and that coronaviruses have evolved in different ways to ensure that this occurs. Recent studies provide evidence that for the SARS-CoV S protein, the most biologically relevant protease may be TMPRSS2. This transmembrane serine protease, which is expressed in pneumocytes, co-localizes with and binds to AC2. In cells expressing TMPRSS2, SARS-CoV enters at the cell surface and is insensitive to cathepsin inhibitors and lysomotropic agents. Just as the mechanism of S protein proteolytic activation is variable, so too is its location. Some coronaviruses, such as most strains for MHV, fuse with the plasma membrane, whereas others, such as TGEV, HCOV229E, and SARS-CoV, can enter cells through receptor-mediated endocytosis and then fuse with the membranes of acidified endosomes. The boundary between these two modes of entry may easily shift. For one strain of MHV, that is MHV4, as few as 3 amino acid changes in the heptide repeat regions of S2 switches the virus from plasma membrane fusion to acid pH dependent fusion. It remains unresolved whether acidic pH as per se is required for S protein conformational changes, or whether this reflects the requirements for activation of endosomal proteases during infection of some types of cells. The coronavirus S protein is a class 1 viral fusion peptide with domains functionally similar to those of the fusion proteins of phylogenetically distinct RNA viruses such as influenza virus, HIV, and Ebola virus, but on a much larger scale. As in those other viral fusion proteins, the coronavirus S2 moiety contains two separated heptide repeats, HR1 and HR2, with a fusion peptide upstream of HR1 and transmembrane domain immediately downstream of HR2. The exact assignment of the fusion peptide is not agreed upon. However, receptor-mediated conformational changes in S1 and the dissociation of S1 from S2 are thought to initiate major rearrangements in the remaining S2 trimer that proceed through multiple intermediate states. These rearrangements ultimately expose the fusion peptide, which interacts with the host cellular membrane, and the two heptide repeats in each monomer are brought together to form an antiparallel 6-helix bundle. The 6-helix bundle is an extremely stable rod-like complex, the biophysical properties of which have been extensively studied. Highly similar crystallographic structures have been solved for the 6-helix complexes from both the MHV S protein and the SARS-CoV S protein. These show the three HR1 helices forming a central coil core some 2 to 3 times larger than its counterparts in other viruses. Arrayed around this, the three shorter HR2 helices in an antiparallel orientation pack into the grooves between the HR1 monomer via hydrophobic interactions. The outcome of the formation of the 6-helix bundle is the juxtaposition of the viral and cellular membranes in sufficient proximity to allow mixing of their lipid bilayers and the deposition of the contents of the virus into the cytoplasm. Following delivery of the viral nucleocapsid to the cytoplasm, the next event is the translation of replicase gene from the genomic RNA. This gene consists of two large ORFs, rep1a and rep1b, that share a small region of overlap. Translation of the entire replicase depends on a mechanism called ribosomal frame shifting, whereby, with a fixed probability, a translating ribosome shifts one nucleotide in one direction from the rep1a reading frame into the rep1b reading frame. This repositioning is programmed by two RNA elements embedded near the region of the overlap that were discovered in studies of IBV. The first element is the 5' UUUAAAC3' heptanucleotide slippery sequence, which is identical for all known coronaviruses and has apparently been selected as optimal for its role. The second element, located a short distance downstream of the slippery sequence, is an extensively characterized RNA pseudoknot structure. This latter component was initially thought to be a classic two-stem H-type pseudoknot. However, recent analysis of the SARS-CoV frame shifting support a more elaborate structure that includes a third stem loop within pseudoknot loop. The two elements act together to produce the co-terminal polyprotein products PP1A and PP1AB. During most rounds of translation, the elongating ribosomes unwind the pseudoknot and translation terminates at rep1 stop codon, yielding the smaller product PP1A. Some fraction of the time, however, the pseudoknot blocks the mRNA entrance channel of the ribosome. The consequent pause required for the ribosome to melt out the mRNA structure allows the simultaneous slippage of the P and A site transfer RNAs into the rep1B reading frame. This results in the synthesis of PP1AB when elongation resumes. Studies of receptor gene expression suggest that the incidence of coronaviral ribosomal frame shifting is as high as 25% to 30%. However, the in vivo frequency in infected cells remains to be quantified. It is thought that the role of programmed frame shifting is to provide a fixed ratio of translation products for assembly into a macromolecular complex. It is also possible that frame shifting forestalls expression of the enzymatic products of rep1b until the products of rep1a have been prepared, a suitable environment for RNA synthesis. Polyproteins PP1A that are 440 to 500 kilodalton and PP1B 740 to 810 kilodalton are autoproteolytically processed into mature products that are designated NSP1 to NSP16 except for the gamma coronaviruses which do not have a counterpart of NSP1. From work begun with early studies of MHV, complete processing schemes have now been solved for all replicases of multiple coronaviruses representing all three genera. Processing also generates many long-lived partial proteolytic products which may have functional importance. There are two types of polyprotein cleavage activity, one or two papain-like proteases or TL-Pro which are situated in NSP3, carry out relatively specialized separation of NSP1, NSP2 and NSP3. The main protease or M-Pro, NSP5, performs the remaining 11 cleavage events. M-Pro is often designated the 3C-like protease or 3CL-Pro to point out its distant relationship to the 3C proteins of the coronaviruses. Several crystal structures have been determined for TL-Pro and M-Pro of SARS-CoV and other coronaviruses, and these enzymes present attractive targets for antiviral drug designs. The processed NSPs reassemble to form the coronavirus replicase, which is also referred to as the replicase transcriptase complex or the RTC. The challenge of designing the roles of many NSP components of the RTC was initially addressed by foundational studies in bioinformatics, which is a discipline that continues to inform the analysis of the intricate molecular machinery. Besides PL-Pro and M-Pro, the products of Rep1A contain several activities that establish cellular conditions favorable for infection. Some of these are directly linked to RNA synthesis. Others are non-essential for viral replication in tissue culture. However, they can have major effects on the virus host interactions. The very first polyprotein product, NSP1, exhibits a broad repertoire of antagonistic activities that selectively inhibit host protein synthesis and IFN signaling. By contrast, NSP2 is completely expendable and as yet has no demonstrated function. NSP3 is by far the largest of the RTC proteins. It consists of a concatenation of individual structural modules that are arranged as globular domains separated by flexibly disordered linkers. At the amino terminus of NSP3 are ubiquitin-like and acidic domains that interact with SR region of the N protein. It is proposed that this interaction tethers the genome to the assembling RTC to allow formation of initiation complex for RNA synthesis. Located within NSP3R1 or 2 PL-Pro modules, in addition to protease activity, PL-Pro domain possess deubiquitinase activity, which forms another part of the viral arsenal that counters host innate immunity. Expression and assembly of the RTC sets the stage for viral RNA synthesis, a process resulting in the replication of genomic RNA and the transcription of multiple subgenomic RNAs. The latter species serve as mRNAs for genes downstream of the replicase gene. Each sgRNA consists of a leader RNA of 70 to 100 nucleotides, which is identical to the 5' end of the genome, joined to a body of RNA, which is identical to a segment of the 3' end of the genome. The fusion of the leader RNA to body RNAs occurs at short motifs. Like the genome, the sgRNAs have 5' cap and 3' polyadenylate tails. Together, these transcripts form a 3' nested set, the single most distinctive feature of the order Nidovirus. Synthesis of both genomic RNA and sgRNAs proceeds through negative strand intermediate.